Three things were found at the scene: a fine rust-colored dust on the underside of a leaf, a web so thin it only appears when the light hits it sideways, and a plant that was healthy last Tuesday and is dying today. Prints. No sound, no visible suspect, only the dust. The dust is not dust. Look closer and each speck resolves into a body. 8 legs, 2 dark eye spots, a translucent shell tinted the color of oxidized iron, copper, or old blood dried on cotton. At full size, the suspect is roughly 400 micrometers long. That is smaller than the width of a human hair is wide. To see one clearly, you need a hand lens. 1,000, you only need to turn the leaf over. Its scientific name is Tetranychus urticae. In English, we call it the two-spotted spider mite, and it belongs not to the insects, as most people assume, to the arachnids, the same ancient class that produce scorpions, harvestmen, and the spiders in your basement corners. It is, quite literally, a spider. A spider so small it can ride the wind. And yet, this creature, which weighs less than a grain of pollen, is considered one of the most economically destructive organisms on Earth. It attacks more than 1,100 plant species. It damages crops across every continent except Antarctica. Greenhouses have been emptied by an infestation that began, invisibly, with a single female arriving on the sole of a shoe. Here is the contradiction. Something this small should not matter. This fragile should not survive our chemistry, our climates, our efforts to erase it. But it does. It has. For at least 65 million years, by molecular estimates, spider mites were already old when the last dinosaurs died. We thought we understood them. Gardeners called them a pest. Farmers called them a nuisance. Chemical companies called them a market. Of the 20th century, the entire relationship between humans and Tetranychus urticae consisted of a single verb: kill. But in the last 30 years, something began to shift in the scientific literature. Corologists, the specialists who study mites and ticks, began publishing papers that did not read like pest control manuals. They read like anthropology. They read like astonishment. The mites, it turned out, are not simple. They were doing things that 8-legged animals 400 micrometers long are not supposed to be able to do. The case had to be reopened. Note the webbing. That fine, ghostly veil across the leaf is not incidental. It is architecture. Spider mites produce silk from glands near their mouthparts, and unlike true spiders, they do not weave to trap prey. They weave to build cities. There creates a controlled microclimate. It raises humidity. It shields the colony from wind and rain. It carries chemical signals, pheromones that tell other mites where to feed, where to lay eggs, and when to leave. Under an electron microscope, the strands are only about 30 nanometers thick, which places spider mite silk among the finest natural fibers ever measured. Researchers in materials science have studied it as a potential model for ultralight textiles. The rust-colored dust on your plant is not just a colony. It is a covered settlement, roofed in silk. Observe the timing. Single female spider mite, under warm conditions, can produce up to 20 eggs per day. Those eggs hatch in as little as 3 days. The hatchlings reach reproductive maturity in 5. Population can double every 72 hours. This means that under ideal summer conditions, one mite arriving on Monday can, by the end of the month, have produced a lineage numbering in the millions. A metaphor. It is arithmetic. It is why a gardener who notices nothing on the 1st of July can lose a tomato plant by the 15th. Cross-reference this. In 2011, an international consortium sequenced the full genome of Tetranychus urticae and published it in the journal Nature. What they found was, in the polite language of scientists, unexpected. Mite genome contains an unusual number of genes involved in detoxification, specifically families of enzymes called cytochrome P450s and glutathione S transferases. These are the same enzymes your liver uses to break down alcohol and medications. The spider mite has more variants of some of these enzyme families than almost any other arthropod ever sequenced. Translation. Creature is a chemist. It can metabolize plant toxins that would kill most insects, and it can develop resistance to human-made pesticides within a handful of generations. In some populations, resistance to a new chemical has been documented in as little as 2 years of exposure. We have been trying to poison something that evolved to eat poison for a living. Note what it eats and how. A spider mite does not chew. It carries a pair of stylets, needle-like mouthparts, which it drives into a single plant cell at a time. It then injects saliva that liquefies the contents and drinks the cell dry. Reception. Each mite empties between 18 and 22 cells per minute. A moderate infestation can drain hundreds of thousands of cells per day from a single leaf. This is why infested leaves develop that characteristic stippled, silvery, then bronzed appearance. You are not looking at damage in the abstract. You are looking at a map of every individual cell that has been emptied. Rust color of the mites themselves comes partly from the pigments they concentrate from those cells. Carotenoids, the same class of compounds that turn autumn leaves red. They are, in a sense, wearing what they ate. The pattern of movement. Spider mites cannot fly. They have no wings. Their legs are built for walking on leaf surfaces, not for long distances. And yet, they are found on remote islands, on mountain summits, on ships far from shore. The mechanism is called aerial dispersal, or ballooning. When a colony grows too crowded, individuals climb to the highest point available, the tip of a leaf, of a stem, and release a strand of silk into the air. The strand catches the breeze, and the mite, weighing less than a snowflake, is lifted. Reception. Field studies have recovered spider mites from atmospheric samples collected at altitudes above 3,000 meters. Some have been documented traveling hundreds of kilometers on a single flight. They are, functionally, a drifting biomass of arachnids suspended in the lower atmosphere at any given moment, waiting to descend on the next available leaf. The rust dust on your basil did not walk there. It fell from the sky. Reference the eyes. A spider mite has 2 simple eye spots, and for a long time we assumed they detected only light and shadow. Recent work suggests otherwise. Behavioral experiments have shown that spider mites can distinguish between wavelengths. They orient toward specific colors of light, avoid others, adjust their egg laying based on the light spectrum passing through a leaf. They appear to perceive ultraviolet, which is invisible to us. And they use these cues to time their diapause, a state of suspended animation in which they overwinter, sometimes for months, tucked into bark crevices or under fallen leaves. They do not simply see. They read the season through the light. Note the final piece of evidence. Spider mites communicate. When one individual is attacked, for example, predatory mite of the genus Phytoceulus, it releases a chemical alarm signal into the silk. Nearby mites detect it and respond by fleeing, dropping off the leaf on silken lines, or entering a defensive posture. Astonishingly, the plants themselves respond. Published in journals of chemical ecology has shown that plants under spider mite attack release volatile compounds, airborne chemicals that attract those same predatory mites. The plant is calling for help. Predator arrives. The mite detect the predator's arrival through the silk and scatter 3 species, 3 languages, 1 conversation unfolding on a leaf you were about to throw away. When I first connected these dots, I genuinely sat back in my chair. I had spent most of my life considering spider mites, when I considered them at all, as a maintenance problem. Something to spray, something to hose off. Rusty stain on an otherwise healthy houseplant. I had not considered that the stain was a civilization, that it wove silk finer than any human textile, that it carried a genome so rich in chemical defense enzymes that we, with our laboratories and our billion-dollar pesticide industries, could not consistently defeat it, that it launched itself into the stratosphere on threads of its own making, that it spoke in molecules, and to the plants it consumed, and to the predators that hunted it. I had considered none of this. And, I suspect, if you are honest, neither had you. The verdict, then, the rusty dust on your plants is not dust. dirt. It is not the beginning of a fungal disease or the residue of hard water. It is a species older than most flowering plants, with a genome more sophisticated than our own for the specific job of surviving what tries to kill it. Weaving a silk city on the underside of a leaf you barely glanced at this morning, you may still choose to remove it. Perception. That is your right as a gardener. But now, at least, you will know what you are removing. What's your verdict? Tell me in the comments. And if you've ever watched a plant collapse in a single week without knowing why, want the full story. I read every reply, case closed. But there is another file already open on my desk. And this one involves a suspect that has been living, undetected, the wood of your window frames, quietly rearranging the geometry of your home in a way that has taken it, on average, 7 years to complete.